Viruses are everywhere. They are tiny infectious agents, far smaller than bacteria. They inhabit every corner of the planet, from the depths of the oceans to the heights of the atmosphere. Virology is the study of these fascinating and often formidable entities. It is a branch of microbiology that explores the structure, replication, evolution, and impact of viruses on life as we know it. Virology plays a crucial role in understanding and combating infectious diseases that affect humans, animals, and plants. Viruses are not considered living organisms in the traditional sense. They lack the cellular machinery necessary for independent existence and reproduction. Instead, they are obligate intracellular parasites, relying entirely on host cells to carry out their life cycle. Viruses hijack the host cell's metabolic machinery, using it to replicate their genetic material and produce more viruses. This parasitic nature makes viruses both intriguing and challenging to study. Virology is an essential field of study because viruses have a profound impact on human health, agriculture, and the environment. Viral infections can range from mild, self-limiting illnesses to severe, life-threatening diseases. Understanding how viruses spread, replicate, and cause disease is crucial for developing effective prevention and treatment strategies.
The history of virology is intertwined with the history of infectious diseases. From the earliest days of human civilization, people have been grappling with the mysterious and often deadly forces of viral infections. Long before viruses were identified, their presence was felt through the devastating epidemics that ravaged human populations. These outbreaks would sweep through communities, leaving a trail of suffering and death in their wake. Ancient civilizations recorded outbreaks of diseases that we now know are caused by viruses such as smallpox, polio, and influenza. These records, often inscribed on clay tablets or written in ancient texts, provide a glimpse into the past struggles against these invisible enemies. These early encounters with viral diseases highlighted the invisible threat posed by these infectious agents. Communities were often left bewildered and desperate, trying to understand and combat these unseen foes. One of the earliest documented viral diseases is smallpox. This highly contagious and often fatal disease has been a scourge on humanity for centuries. This highly contagious and often fatal disease left its mark on history, shaping civilizations and influencing the course of empires. The impact of smallpox was so profound that it altered the demographic and social structures of entire regions. Smallpox epidemics decimated populations, leaving behind a trail of death and disfigurement. The scars left by smallpox were not just physical, but also deeply psychological, affecting the collective memory of societies. The ancient Egyptians, Chinese, and Indians all developed methods of variolation, a precursor to vaccination, in an attempt to control smallpox outbreaks. These early medical practices were rudimentary, but represented significant strides in the fight against viral diseases. Variolation involved exposing healthy individuals to material from smallpox pustules, hoping to induce a mild infection that would confer immunity. This practice, though risky, laid the groundwork for the development of modern vaccines. Another ancient viral disease is rabies. This terrifying disease has haunted human and animal populations for millennia. This deadly disease, primarily transmitted through the bite of infected animals, causes inflammation of the brain and spinal cord. The symptoms of rabies are harrowing, often leading to a painful and inevitable death if left untreated. Rabies has been recognized since ancient times, with Babylonian tablets dating back to 2300 BC mentioning the disease. These ancient records show that humanity has long been aware of the dangers posed by rabies. The ancient Greeks developed the first recorded treatment for rabies, cauterizing the wound with a hot iron in an attempt to kill the virus. This method, though primitive, was an early attempt to mitigate the effects of a viral infection, showcasing the enduring human endeavor to combat these deadly diseases.
The late 19th century marked a turning point in the history of virology. Scientists began to unravel the mysteries of these elusive infectious agents. The development of the porcelain filter, a device capable of removing bacteria from liquids, played a pivotal role in this breakthrough. In 1892, Russian botanist Dmitry Ivanovsky made a groundbreaking observation while studying tobacco mosaic disease, a disease that stunted the growth of tobacco plants. Ivanovsky discovered that the infectious agent responsible for tobacco mosaic disease could pass through the porcelain filter, suggesting it was much smaller than bacteria. He called this infectious agent a filterable virus, marking the first time the term virus was used in its modern context. Ivanovsky's work laid the foundation for the field of virology, demonstrating the existence of infectious agents smaller than bacteria. Building upon Ivanovsky's work, Dutch microbiologist Martinus Beijerinck conducted further experiments on tobacco mosaic disease. In 1898, Beijerinck independently confirmed Ivanovsky's findings and introduced the term contagium vivum fluidum to describe the infectious agent. He proposed that this agent was a new type of infectious particle distinct from bacteria that could replicate only within living cells.
Section 4. The tobacco mosaic virus, a turning point in virology. The tobacco mosaic virus, or TMV, played a pivotal role in the early development of virology. This virus, which primarily affects tobacco plants, was one of the first viruses to be discovered and studied in detail. It became a model organism for studying viruses, allowing scientists to delve deeper into their structure, replication, and transmission. Researchers were fascinated by how such a small entity could cause significant disease in plants. In 1935, American biochemist Wendell Meredith Stanley achieved a groundbreaking milestone by crystallizing TMV. This was a monumental achievement in the field of virology and biochemistry. Stanley's work demonstrated that viruses, despite their ability to replicate and cause disease, could be crystallized like inert chemicals. This was a revolutionary concept at the time, challenging existing notions about the nature of viruses. This discovery blurred the lines between the living and non-living worlds, raising fundamental questions about the nature of life itself. Scientists began to ponder whether viruses were living organisms or merely complex molecules. Stanley's crystallization of TMV earned him the Nobel Prize in Chemistry in 1946 and opened up new avenues for studying viruses using the tools of biochemistry and biophysics. His work laid the foundation for modern virology. The structure of TMV, elucidated through X-ray crystallography studies, revealed its simple yet elegant design. This method allowed scientists to visualize the virus at an atomic level. TMV consists of a single strand of RNA, the genetic material, encapsulated within a protein coat. This RNA strand carries all the information needed for the virus to replicate and infect new cells. This protein coat, made up of multiple identical protein subunits, protects the viral RNA and facilitates its entry into host cells. The interaction between the protein coat and host cells is crucial for the virus's life cycle. The simplicity of TMV's structure has made it a valuable tool for studying virus assembly and disassembly. By understanding these processes, scientists can develop strategies to combat viral infections in both plants and animals.
Section 5. The rise of molecular virology. DNA, RNA, and the genetic code. The mid-20th century witnessed a revolution in biology with the discovery of DNA as the carrier of genetic information. This pivotal moment marked the beginning of a new era in scientific research, transforming our understanding of life at the molecular level. This discovery had a profound impact on virology, leading to the emergence of molecular virology. Researchers began to explore the intricate details of viral structures and their genetic material, uncovering the secrets of how viruses operate and interact with their hosts. Scientists realized that viruses could have either DNA or RNA as their genetic material, further expanding the diversity of the viral world. This revelation opened up new avenues for research, as scientists sought to understand the implications of these different types of genetic material on viral behavior and evolution. The development of techniques to study and manipulate viral genomes allowed researchers to unravel the molecular mechanisms underlying viral replication, gene expression, and pathogenesis. These advancements provided critical insights into how viruses replicate and cause disease, paving the way for the development of new antiviral therapies and diagnostic tools. One of the key discoveries in molecular virology was the identification of reverse transcriptase. an enzyme that allows RNA viruses to convert their RNA into DNA. This enzyme plays a crucial role in the life cycle of many viruses, enabling them to integrate their genetic material into the host's genome. This groundbreaking finding, made independently by Howard Temin and David Baltimore in 1970, revolutionized our understanding of viral replication. Their work demonstrated that the flow of genetic information could be more complex than previously thought, challenging existing paradigms in molecular biology. Reverse transcriptase is essential for the replication of retroviruses, a family of viruses that includes HIV, the virus that causes AIDS. The ability of these viruses to reverse transcribe their RNA into DNA allows them to persist in the host and evade the immune system, making them particularly challenging to treat. The discovery of reverse transcriptase had far-reaching implications for both virology and molecular biology. It not only advanced our understanding of viral replication, but also provided new tools and techniques for studying genetic material. It provided a mechanism for the flow of genetic information from RNA to DNA, challenging the central dogma of molecular biology, which stated that genetic information flowed only from DNA to RNA to protein. This new understanding prompted scientists to rethink the fundamental principles of genetic information transfer. Reverse transcriptase became an essential tool in molecular biology, enabling techniques such as reverse transcription polymerase chain reaction, or RT-PCR, which is widely used to detect and quantify RNA molecules. This technique has become indispensable in research and clinical diagnostics, allowing for the precise measurement of gene expression and the detection of viral infections.